Hello everyone, myself Dr. Bhagwan Lal Kalal, Assistant Professor, Department of Chemistry, IPS Academy, Indore. Today we are going to determine dissolved oxygen by Winkler method. So let's start with the introduction. First thing is that what is dissolved oxygen? Dissolved oxygen is the oxygen like oxygen in an environment. The difference is that it is the dissolved oxygen in water. And DO is a measure of how much oxygen dissolved in the water. That is available oxygen for aquatic organism. It is necessary to survive aquatic life, that is plants and fishes and other organisms. The other thing, which factor decide the dissolved oxygen, how much oxygen will found in water. Overall, two type of, by two way, oxygen, amount of oxygen is varied. First, the source of oxygen in water. In this category, we can add 2 points: aeration of water, that is movement, any type of movement in water, like falling of lake, falling of water in lakes, that will increase the amount of oxygen in water. Other factor that will increase the dissolved oxygen in water is photosynthesis by aquatic vegetation. That is, as we know, that when algae and other aquatic plant, when synthesize food by photosynthesis, they will generate oxygen. This oxygen is dissolved in water and available as dissolved oxygen. The other factor that will decide the amount of dissolved oxygen in water is the consumption of dissolved oxygen. As we know, all type of organism will use oxygen in their respiration. Plant as well as fishes and other animals that found in water uses oxygen, that result decrease in DO in water. Other factor that will affect the quantity of dissolved oxygen is eutrophication. The next thing is, what is eutrophication? That will eutrophication is termed as gradual increase in concentration of phosphorus, nitrogen and other plant nutrient in aging aquatic ecosystem such as lake. The productivity or fertility of such an ecosystem naturally increase as the amount of organic material that can be broken down into nutrient increase. It means when we dump nitrogen or other element as a pollutant in water body, it will increase the algae and other vegetation in water body. When these degrade, then naturally the nutrient in the water body will increase. This process is called, this increase in nutrient in water body is called eutrophication. It is the process of increase in nutrient in water body by degradation of animals and plants, algae, etc. Once again, which factor affect the DO, that is dissolved oxygen? So that is, many factors will decide the amount of dissolved oxygen in water. First thing is salt concentration. When salt concentration increase in water, that will decreases the dissolved oxygen. Similarly, increase in temperature also decreases the DO. It is due to the thermal vibration of water molecule, which is the removal of dissolved oxygen from water. Other thing is biological activities, like when the plant, algae or fishes population increases in water, that definitely then they will use as DO, dissolved oxygen in water. Ultimately, dissolved oxygen in water will decreases with the increase of population of fishes and other aquatic animals. Other thing is the factor that decide the DO is the depth. From depth of water body, as there is a plenty of oxygen in our environment, almost 21%. This oxygen is diffuses in water. Ultimately, at the surface, concentration of dissolved oxygen is always higher. Other thing is that plenty of light is available on the surface of water, that results more photosynthesis at surface. It will eventually increase the DO at the surface. Other thing is when the water is falling in water body, that will cause disturbance in water surface and formation of bubbles. When water drops from height, it will eventually increase the dissolved oxygen at the surface, and at the depth this disturbance will less. And other thing is the amount of light is also decreases with depth, that will, due to which the rate of photosynthesis will decreases, and eventually dissolved oxygen will also occur available less. Next, as we are going to determine the dissolved oxygen, why we are going to why dissolved oxygen is measured. There are 3 or more reasons. That's why we determine the dissolved oxygen. First, dissolved oxygen tell us how much water body can support life. It means if there is a less or dissolved oxygen, then aquatic animal, microorganism that can't survive, as oxygen is required for everyone. When dissolved oxygen will be less, then this organism will die because they will be unable to respirate. Other thing is dissolved oxygen is an indicator of health of water body. Means higher the dissolved oxygen, more healthy will be the water body, and less dissolved oxygen is an indication of poor health of water body. Other thing is dissolved oxygen ensure. Dissolved oxygen is a measure that how much oxygen available for microbes to convert for their activity. As you know, microbes in water convert the organic waste into harmless end product. So it is tell us that water body can support the life of microorganism or not. As microorganism is required positively to convert wastewater into harmless end product. Now Let's see some ideal parameters or warnings regarding DO parameters. That is, when the dissolved oxygen is less than 0.2 ppm, then water body is termed as anoxic, means there is no oxygen that can support the life. Dissolved oxygen is measured in milligram per liter. That is also considered as concentration in ppm. Next, fishes in water required dissolved oxygen over 3 ppm to survive. Below 3 ppm, very difficult for them to survive. In seawater, at 100% saturation, at 20 degrees centigrade, the dissolved oxygen ideally found is 9.30 ppm. And in healthy water body, when we measure DO, it is found in range between 6.58 milligram per liter. There are two methods by which we can determine dissolved oxygen. First one is Winkler method. It is a volumetric method. And second one is electrometric method, in which we use membrane electrode method, for which make the basis for Winkler method. In this method, we have to fix the dissolved oxygen present in water at the sampling site, as with the time and with the movement in water, quantity of dissolved oxygen varies. Therefore, it is very favorable, ideal that we determine the dissolved oxygen at the sampling site, or otherwise we can also fix the quantity of oxygen by reagent, and then bringing the sample at our lab, we can determine the dissolved oxygen. So first, take the water sample in BOD bottles, and then we add some reagent to fix the dissolved oxygen, so that the amount of dissolved oxygen remain fixed. Fixing of dissolved oxygen results orange brown PPT. These PPT then dissolve in acid. Reaction of sulfuric acid produces iodine, and this iodine which is formed is in proportion of dissolved oxygen present in water. Then the produced iodine is determined by titrating the reaction mixture with standard hypo solution. When iodine complex with starch indicator, it will produce violet blue color. So when we titrate with hypo by addition, this concentration of this complexes decreases as it decomposes, and near end point the blue color decolorizes. When it completely decolorizes, it is an indication of end point, and the amount of hypo that is used for decolorization of blue color will determine the quantity of dissolved oxygen. Dissolved oxygen analysis is best done in the field as sample will be less altered by atmospheric equilibrium. Now let's see the chemistry behind the Winkler method. For fixing, we using the manganese sulfate solution. The dissolved oxygen present in water will turn manganese ion into manganese dioxide precipitate. The color of precipitate is yellow brown. These manganese dioxide precipitate, when added sulfuric acid and potassium iodide, manganese dioxide turns the potassium iodide, mean iodide ions, into iodine. The produced iodine will be proportional to the amount of manganese dioxide that is generated by the reaction of manganese ion and oxygen. The produced iodine then estimated by titrating the mixture with hypo solution. In this titration, 5 ml of 0.05 m hypo solution will be equivalent to 1 milligram of dissolved oxygen. See once again the chemistry. We are using manganese sulfate solution for fixing the oxygen. In alkaline condition, it will generate manganese dioxide in yellow brown PPT. This manganese dioxide then is used in the reaction to form iodine by iodide ion. This iodine then we determine by titrating the reaction mixture with hypo solution. In titration, 5 ml of 0.025 N hypo equivalent to 1 milligram of dissolved oxygen. There are some ions that can interfere in the Winkler method. These ions have similar property like dissolved oxygen. Like dissolved oxygen, that can also produce iodine in the reaction. Nitrite and sulfite ion generate iodine like dissolved oxygen. When it also produces iodine, then the result that we occur will be wrong by the presence of this ion. So before determining dissolved oxygen, we have to destroy these ions, and these can be easily removed by addition of sodium azide and alkali Ki solution. How they are eliminating these ions? When we add sodium azide, it will decomposes nitrite ion, which is found in the form of nitrous acid, into nitrogen. Nitrous oxide is the equation of this reaction. You can clearly see that sodium azide reacting with nitrous acid and generating nitrogen and nitrous oxide. The next is reagent preparation that is required for the measurement of dissolved oxygen. We have required 4 solutions. First one is manganese sulfate solution. It is prepared by dissolving 36.4 gram manganese sulfate in 100 ml water. The other one is alkaline iodide sodium azide solution. It is the mixture of KOH, sodium azide and Ki. We have to take 70 gram of KOH, 15 gram of Ki and 1 gram of sodium azide. First dissolve these quantities in water, and then we have to make up the solution 100 ml with boiled water. The next solution that is we have to prepare is hypo solution. For this it is required for titration purpose. We have to prepare 0.025 and sodium thiosulfate solution. To make this solution, we have to dissolve 25 gram of hypo and 100 gram of NaOH in water, and then make the volume 100 ml. Next is starch indicator. To prepare it, we have to dissolve 2 gram starch in 100 ml water and then boil it. Procedure of Winkler method. For that, we have to for collection of water sample, we have to go in the field. There we have to fill the BOD's bottle. The capacity of BOD bottle is 300 ml. We have to fill bottle completely so that no environmental oxygen capture or remain in the bottle. We have to take precaution that no bubble formation take place when we collect the water sample. After collection of water from the field, immediately we have to add 12 ml of manganese sulfate solution. This addition will fix the oxygen present in water in the form of precipitate. While we adding the manganese sulfate solution, we have make precaution that the reagent should be added in BOD bottle just over the surface so that no oxygen from air can dissolve in bottle. Next, we have to add 2 ml of alkali iodide azide reagent in same manner so that no oxygen dissolve in water by environment. Then we have to stopper the bottle so that no air introduced from the environment. Then mix the sample by inverting the bottle several times. If any air bubble formation take place in bottle, then throw the entire mixture and repeat the step of collection of water sample. If oxygen is present a brownish orange precipitate will generated in the bottle. When this flocks settle, again we have to shake the bottle by turning it and mix the sample several times and stand it for settling again. Now you can determine the dissolved oxygen at the field or by taking the sample in lab. In next step, we have to add 2 ml concentrated sulfuric acid in a manner that no oxygen dissolve from the environment to sample. This mixture can be stored for 8 hours if kept in cool condition and dark places. Now take 201 ml of reaction mixture from BOD bottle to a glass flask and titrate by adding hypo solution till the reaction mixture turn pale straw color. Now add starch indicator. It results blue color due to the formation of starch iodide complex. Continue the adding of hypo solution till the blue violet color discharges. The discharge of blue color is an indication that the end point is reached. The amount of hypo that is used for decoloration of violet blue color will be directly proportional to the oxygen present in water. In practically, the parameter is set in such a way that the amount of the volume of hypo that is used in reaction will directly indication of dissolved oxygen. That is, if 5 ml hypo is used in the reaction, that will give the result 5 ppm dissolved oxygen. It means the volume of hypo that is used in reaction will give directly the amount of dissolved oxygen in milligram per liter. Now we are going to our lab and practically we will determine the dissolved oxygen of a water sample, that is we have already collected from our nearby water body.